Okay, so today we are looking at the structure of cell membranes as well as membrane transport. So a little bit of everything going on today. What you need to do is first, if you've not already done so, there is a link to a SurveyMonkey survey. After you have done that, I have guided notes for you on this section. So you can either print them out or you can do them directly on the computer, just because I know printing isn't necessarily something y'all can do. So as I'm going through this, I am going to have this off to the side on my screen and hopefully I'll hit everything. And if you've got questions, as always, let me know. Our big ideas for today, really three of them: where are membranes found, and what are the structures of them? Those two pieces, those two questions that you should be able to answer by the end are very much like knowledge-based, and it's straight out of the book. We're also going to be looking at the function of membranes, and this one is one that is a little bit of more of a foreshadowing for the rest of the unit. So let's start out by looking at where membranes are going to be found. Membranes are going to be found around the outside of both prokaryotes. That's these guys here. Oh, I need a bigger pen. There we go. These guys here, as well as all of that. And that's going to be the one that we call the cell membrane, the one that goes around the entire thing. And so we know that all cells are going to have a cell membrane, both the prokes, which are going to be things like your bacteria, as well as the eucs, which are going to be your all of your different multicellular organisms, so plants, animals, fungi, as well as protists, which are like those weird little unicellular eukaryotes. Now, if we're looking at the prokaryotes, remember that they're defined as not having any membrane-bound organelles. Their DNA is just floating in the cytoplasm all loosey-goosey, but when we switch over to the eukaryotes, they do have this nuclear envelope that surrounds the DNA. In addition, they also have this whole network of all of these folded layers, right? And we've looked at these already, and a whole bunch of little vesicles here, there, and everywhere else, and that all is going to make up the endomembrane system. So this endomembrane system is a really complex, convoluted network that is going to include the endoplasmic reticulum, the Golgi, all these different vesicles, and in plant cells, the large central vacuole as well. Remember, that's going to be where we are making and transporting different proteins throughout the cell, as well as taking things in and moving them around. Then finally, remember that eukaryotic cells also have mitochondria. Those are our powerhouses of the cell, and those have the membrane that wrap the outside of the mitochondria, as well as the cristae, or the kind of those inner folds that make up the area where you're going to have the electron transport chain happening. Hey, look at that, we're tying it back. And then in the plant cells, you're also going to have membranes that are on the outside as well as on the inside of the chloroplast. So the outside being the chloroplast membrane and the inside being the thylakoid membranes. So we're going to find them all over the place. And so now we're finally getting to learn what those membranes are. We've been talking about membranes since basically the beginning. Now time to look at what they are. And there's going to be two of our macromolecules that are really abundant in the membranes. So different sugars are not going to necessarily be as important here. So our carbohydrates are not going to be as integral as two of the other ones. Our nucleic acids are obviously all up in here, so they're not going to be as integral, but we will have two, the lipids and the proteins, that are particularly abundant within the cell membranes. So let's go ahead and start by doing a quick review of what we remember about lipids. A lipid, for the most part, your fats, oils, and waxes as well, are going to have this structure right here. We have the glycerol, which is kind of that backbone that then can attach three fatty acids. And if you're remembering, we talked about the dehydration reactions that are going to be able to attach them together. So that dehydration reaction is happening right here. But in the case of in the case of membranes, these lipids are a little bit modified. So instead of having that third fatty acid right here, we're going to have a phosphate group. And what this is a really polar piece. Remember that the phosphate group is a phosphorus and a whole bunch of oxygens, and other components. And so this phosphate group right here is going to be what we call really hydrophilic. It is water loving, it is polar. So all of the negative oxygens, right, they're very electronegative, if not straight up negatively charged, then those are all going to very strongly attract to water. The fatty acids down here are long hydrocarbons, carbon, 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 a bunch of hydrogens. All of those nonpolar bonds means that overall the lipid tails are nonpolar. They're not going to associate well with water. Okay, so here is our typical model of a lipid, but in the case of phospholipids, that third one that would be right here is like nah, and we're getting this phosphate head. So we've got the phosphate head, the lipid tails, and what we'll see is they orient themselves in such a way that the hydrophilic head is always facing water and the hydrophobic tails are always sheltered away. And the best way to do this is to form what's called a bilayer, or two molecules thick layer. So the phospholipid bilayer is going to form with the hydrophilic heads pointing towards the water and the hydrophobic tails on the inside. And so maybe this is the outside of the cell and this is the inside. Okay, making sense so far? So the majority of the cell membrane is going to be that phospholipid bilayer, that two layer of these molecules that form together to make kind of like an oily membrane, obviously, but like a saran wrap type of texture. Okay. In addition, we're also going to have a whole bunch of proteins that kind of get embedded into the membrane, and they might just be on one side or the other side, either the inside or the outside layer, or they may go the entire way through. We're going to call these guys that are only on one side the peripheral proteins, and the ones that go the entire way through integral. And so a quick reminder about proteins when we're looking at them, they're the long strands of amino acids. Those amino acids are going to fold. And remember that there's 20 different amino acids. Each one has its own special chemical characteristics. So what we find is that amino acids, maybe right here, tend to be really nonpolar. And that's going to allow them to interact with the nonpolar tails of the phospholipid bilayer, whereas parts right in this area and this area tend to be really polar. So their polarity, their amino acids, like the chemical structure, or sorry, the chemical characteristics of those different amino acids are what are going to determine how this whole protein folds and kind of lock it into the cell membrane. Let me go back up here. When we're looking at different membrane proteins, they're going to have just as many functions as what we found with proteins before. Structurally, they're going to be the attachment point both for other cells as well as the cytoskeleton. They can play roles in cell communication, so picking up messages from the outside and delivering them into the cell. They can be really important for anchoring. Did I already say that? I think I already said that. They can be enzymes, and what's really important here is that they can also act as kind of channels, gateways between the outside of the cell and the inside, so things that might not be able to get directly through the membrane or through the phospholipid bilayer at least can get through the membrane, so to either get out of the cell or to enter. A little bit of a COVID link here for you. The proteins that are on the cell membranes are also going to be really important for recognizing different viruses and bacteria and other invaders into our bodies. So if we were to kind of zoom in and let's pretend that this is our cell, the cell membrane is the black line, just pretend it's 2 layers thick, and we've got these little proteins kind of all up in here, embedded in here, and they do a whole bunch of different things that are based on being able to communicate and develop into the certain type of cell that it is. And one type of those proteins that you're going to find are these little recognition sites. And if they bind to some particle, it says to the cell, hey, wait a minute, there's something bad going on. I recognize this thing as something that shouldn't be in the human body or that shouldn't be around me. Go into panic mode, and that's how our immune system works, by recognizing these things that shouldn't be there. And then all of a sudden this guy is like, all right, time to start proliferating, time to make 1,000,000 of me to make an army so I can fight off whatever this guy is. And so that's why our bodies take a while to be able to respond when we have the first encounter with a new virus or any kind of infection, right? The first time, it's going to take about 8 days because we have to first recognize that guy, which is going to have to do with the proteins on the cell membrane being able to go, there's something fishy here. And then from that, they've got to divide and make an entire army. Anyway, good story. So now let's really get into the membrane function. What is the purpose of the membrane? And importantly, it's separating what's going on inside from outside. But it can't just carte blanche block everything from coming in or out. If we go back to our analogy of the city or the town and each of the houses within the city and town, it's not enough just to get materials to the house. You need to get them in the house. In this case, it's not enough to just get oxygen to a cell. You need to get it into the cell where that cell can utilize it. And that means crossing membrane after membrane after membrane. And so the membranes are going to be what we call selectively permeable or semi-permeable, right? And what that means is that they're not just letting everything through, but they are going to be letting some things through. Why would this matter? Well, oxygen's a prime example. You bring in oxygen into your lungs, it gets into your red blood cells, and then from there, the red blood cells are going to move to the body, and when the concentration of oxygen is too low, that oxygen is just going to move through this cell membrane, out of the red blood cell, through the cell membranes of the cells that make up the capillaries. So that tube that acts as our blood vessels are actually made out of cells upon cells upon cells. And so anything that moves into or out of the blood has to move into and out of cells, and therefore through these different membranes, and then into the cells of the different body parts, into the mitochondria. If you're a eukaryotic cell, that's going to make it much more, many more steps to get things from point A to point B, until finally you can get into the area where you're being used, in this case the matrix, where the Krebs cycle is going to happen. You remember that, right? Krebs cycle is happening. Oxygen is going to be needed for the electron transport chain, and that's going to make the ATP. I'm guessing you've blocked that out of your memory. I don't blame you. It's okay. But we'll come back and see it this unit, so it's going to be kind of important. Anyway, so the ability of these cell membranes to act as a semi-permeable barrier is going to be how we regulate what's happening with the cells, and therefore regulate our homeostasis. Okay, I want to do a little bit of a preview of the rest of the unit. So far, everything that I've gone over is pretty much out of chapter 4.2. I'm not going to be doing a ton of extra lectures, and if I do, they're going to be very short. The idea between all of them follows this basic concept, okay? We're looking at how materials either move through the membrane in one direction or the other, and into and out of isn't so much of a big deal. What we're going to focus on really is, are the materials flowing from where there's more to less or less to more? Let's start out by looking at concentration then. When we talk concentration, we're talking about parts out of the whole, right? If you have a very highly concentrated mixture of Kool-Aid, you have a ton of sugar mixed into a smaller amount, versus a weaker Kool-Aid has a lower concentration, and that's going to be a lesser amount or a greater amount of solvent, right? So we have our solute that's dissolved into our solvent. So when we have a high concentration, that either means a really high solute or a really low solvent, or both. And on the flip side, when we have a low concentration, that's opposite. Now, why are we talking about that? As it turns out, most particles move based on their concentration. And we're going to call, when we have one area with a lower concentration and one side with a higher concentration, right? So there's not the same amount on each side. We're going to call that a gradient. And in general, molecules are going to be moving in all directions, completely at random, bouncing off of each other. I've given the analogy in the past of little kids running around in a gym, and I wish so badly right now that my kids had a big old gym to run around in because they are going crazy. But if you can imagine a big gymnasium, right? And here's the door to the gym. And we take a whole bunch of little kids and just let them loose. Now, this is like November 1st, right? Day after Halloween type kids. They are completely juiced up. They are going nuts, bonkers, running all over the place. And at any given moment, any one of them is likely to be just about anywhere. If you open this gym to everybody, they're going to spread all over the place and be bouncing around like crazy kids. And at any given point, maybe Jimmy's over here, maybe Jimmy's over here. It's not that Jimmy himself is going to be moving from a high to a low concentration, but in general, the kids are going to spread out, starting where they're very, very condensed, and then find new areas until they've reached what is called equilibrium, where they're all equally spread out. They're still moving, but they're pretty fairly well spaced. That's how molecules are going to be moving. They're going to move from where they're very, very tightly packed in with others like them to where they're more spread out. Making sense so far? Doesn't matter. You can't answer me. If it's not, make sure to email me. And so when we talk about membrane transport, there's two things that we're going to look at. We're going to look at passive transport first, and that's going to make up the bulk of our conversation, and then active transport a little bit as well. The difference between them is all about which direction molecules are moving compared to the concentration gradient. In passive transport, we're going to have molecules move from an area of high concentration to low. And we're going to call this down the concentration gradient, right? So from high to low. It's those kids just spreading out. And the only energy that's needed is the molecular energy that's already present. As you can imagine, heat up something and it bounces around more and is likely to reach equilibrium faster, right? That's why if you mix Kool-Aid with really, really hot water, it dissolves faster because now these particles aren't just moving a little bit, they're moving like crazy. So passive transport means we're moving material across the membrane from an area of high to low concentration. So if we could picture that there's this barrier here, the cell membrane, if they're going from the side where there's more to the side where there's less, that's passive. It doesn't matter in or out. It just means high to low, okay? On the flip side, picture it's the end of gym and that teacher needs to start corralling all of those kids together and pushing them back into the very crowded hallway, right? They're all nice and spread out here, but we've got to kind of condense them into one area. And that's going to be called active transport, when we take molecules and move them against the concentration gradient from where they're pretty well spread out at a lower concentration to now where there's a greater amount of them or a greater concentration. And as you can imagine, that's going to be something that takes a ton of energy. And what do you think that energy is going to be? Well, we saw this already. ATP, woo woo. So that adenosine triphosphate, that ATP, that battery of the cell is going to power different proteins that are found on the membrane to just start pumping them out, pumping them against the concentration gradient. And so instead of reaching equilibrium, in active transport, we're going to establish a concentration gradient. So we move away from equilibrium. We really kind of push everything in one direction and get it to where it's really concentrated on one side and not so much on the other. Okay, let's put this in context of homeostasis and the multicellular organism's ability to maintain homeostasis, because a lot of the different things that we need, whether it's a nutrient that we're taking in or it's getting rid of a waste, a lot of them are going to rely on passive transport. So as we're looking at passive transport, there's going to be 3 topics that we look at. And for those 3 topics, I'm going to relate each of them to a type of feedback mechanism where homeostasis is regulated. So 3 types are diffusion, facilitated diffusion. Oops, out of room here. And osmosis. Okay, so those are all about how material can get across the membrane. But in all three cases, we have material moving from high to low concentration. Yeah, I feel like I was going to add more to that and then I didn't. On the flip side, when we look at active transport, there's going to be two types that we look at, and these are going to be more important for specialized parts of the body, not necessarily the homeostasis per se, but more specialized functions. For example, our immune system, our nervous system, and so forth are going to rely on certain types of active transport. Really, all of our body does, but not the ones we're looking at for homeostasis. And this is going to be molecular and bulk movement. Okay, so let's kind of wrap up by bringing it back to what we talked about at the beginning. If this represents a multicellular organism, we know that the body has a whole bunch of different areas where stuff can come in or stuff can go out. So going over those again, this is where you should take a second and be active in it. See if you can predict what I'm going to say. Pause if you need. But we've got the digestive system, the respiratory system, the excretory system. And so the digestive system is going to be bringing in glucose and other things. The respiratory system is going to be bringing in oxygen and getting rid of carbon dioxide. The excretory system is going to be getting rid of different nitrogenous wastes. So what's actually happening here, I should have done that. Let's do this. Okay, is let's take, for example, the digestive system. We're going to eat a meal. Our glucose levels inside the digestive system is going to go really high. Our circulatory system is what's going to deliver that material, but the way that glucose is going to get from inside the digestive system to inside the circulatory system is just by moving from where there's a lot of it to where there's less of it. So if we have a ton of glucose in our intestines, in our stomach, then that can kind of seep out from where there's more to where there's less until it gets into the cardiovascular system, into the blood, into the circulatory system, right? So here's our cardiovascular system that just connects everything. And as you can imagine, this cardiovascular system is then going to have a higher concentration than the body cells. So let's say here's a little cell right here. We can say the concentration, concentration of glucose is really, really, really high in the digestive system, and then it's going to diffuse out and be kind of medium in the circulatory system, but it's going to be really low in the body cells, and that's because glucose is constantly being used. And so that's going to allow the circulatory system, the materials there, such as glucose, to move into those body cells. And as it's used, eventually we saw that with any kind of passive transport, eventually what you reach is the state where everything's all at equilibrium. Well, that means that there's no more net movement. So what happens when there's no more net movement? And subsequently, everywhere is using this glucose, so you run out of glucose. So what do we have to do? Well, our digestive system refills up, makes a really high concentration of glucose in the digestive system, spreads it to the circulatory system, that moves it to the body cells, that all gets used up, and as you can imagine, that's going to lead to this constant up and down of glucose availability. Homeostasis, right? Being able to give some kind of negative feedback to say, okay, glucose levels are too high or oh, they're too low. Having a way to regulate that so it stays kind of even though we're going to have times when the glucose is really, really high in the intestines and other times when it's really, really low, we have to keep it available to the cells at all times. How we do that is going to come back to that negative feedback. I'm hoping this is all making at least a wee bit of sense. If you've got questions, just like always, shoot me an email and Happy Tuesday.